I didn't even say hi yet. You're going to start it right away. So, yeah, we have quite a few things to go over, and then I want to spend some time kind of going over the exam so you guys have time to ask questions if you need to when you get your exams back. So I have both lecture and lab exams all graded. Oh. So that will be given back to you guys.
So you had a fun day yesterday.
I couldn't do it yesterday because I am teaching and I have my other classes to teach, so I pretty much did it today. I did a little bit of it yesterday during my break. That was my break. And then today, right after a dentist appointment, I went home and I started trying to finish grading. And then I was over here finishing everything at the last minute. So, yeah. It's a busy semester, but I don't know. I'm trying to keep up because if I fall behind, you guys know what it's like when you fall behind, right? It sucks. So I'm trying not to let that happen. So, on kind of like the bottom of page 26, we have the neuron structure. So we have two of these models. We went over this in lecture, going over the parts of just a basic structure of a neuron. So this is one neuron, but it's like sectioned in half. So we have like the cell body over on this side over here, and then we have the axon over on this side. But like this is connected to this. Does that make sense? So cell body is all of this over here, which is one of the things you want to know. These gray parts that stick out, these are the dendrites. So again, dendrites are going to be the gray parts that are sticking off of the cell body. They're gray on this, not orange. These orange things over here are something totally different. These are going to be the telodendrites or the axon terminals of another neuron that's sending impulses to this neuron. So it's the gray parts that are going to be the dendrites. So dendrites and cell body. This is the nucleus of the cell body. So it's the round part in here. We have these blue dots inside of the cell body. Those are the Nissl bodies, which I think is listed here. Yeah, so that's that fourth term on there, Nissl bodies. Where would you find the nucleolus?
Inside the nucleus.
Inside the nucleus. So that's the purple color thing in there. Inside of the nucleus. Nissl bodies are ribosomes. So it's like clusters of ribosomes. So protein formation or protein production. All the blue dots inside of the cell body are Nissl bodies. Dendrites already mentioned. Axon is this right here. So it's the center line that's going all the way down. We also see it over here as well. So axon is going to be this gray portion, which is the same as this gray portion, or it looks kind of orange with the section. It's going to go all the way down. That's going to be the axon. Okay. If I were to point at, for instance, this layer out here, then that's them looking at the myelin sheath. So myelin is going to be at multiple layers that wrap around or surround the axon.
What did you say was myelin sheath?
Myelin sheath is going to be the multiple layers that's surrounding the axon. But you want to be kind of careful with that. So like on the sides over here, we see multiple layers. That's going to be the myelin sheath. Multiple wrappings around the axon. But if, for instance, if I have a sticker on this pink nucleus over here, that's actually going to be the nucleus of the Schwann cell. So then that's going to be the Schwann cell rather than the myelin sheath.
What do you call it?
So it's a pink part. The pink one, number 7.
That's Schwann cell.
So that's going to be the Schwann cell.
And then the nucleus of the Schwann cells in the
And so the pink is going to be the actual cell, and the darker pink in there is going to be the nucleus. So, so you have neurolemmacyte or Schwann cell. That's going to be if I were to tag number 7, the pink. That would be the Schwann cell. But if I'm tagging like the multiple layers of white surrounding the axon, that would then be the myelin sheath. So that's going to be the difference between the two. We also have axon hillock. So this orange portion right there is the axon hillock. That's basically like where the cell body begins to taper and becomes a little bit more skinny, a little bit more narrow to then lead into the axon. So this orange portion is the axon hillock. Again, that's like the tapering part of the cell body, which eventually leads into the axon. And the last thing is endoneurium, which we didn't talk about yet. But endoneurium is going to be this gray layer right here that is surrounding the nucleus, or the neuron. So like the white multiple layers is going to be the myelin sheath, and then surrounding that over here is going to be the endoneurium. So we'll talk about endoneurium more later on.
Wrap all the way around the myelin sheath, or just in certain spots?
It wraps all the way around.
Okay.
So the whole thing is going to be wrapped around by the endoneurium. So think of it as like a layer of connective tissue, kind of like to keep things in place. That's going to be endoneurium. So endoneurium means like inside, endo, inside, which basically means that we're also going to see perineurium and epineurium when we talk about nerves. So this is all you need to know for now, for today, for the neuron. And then we can move on to the rest of page 26 for brain structures. It basically says that the terms on page 27, and basically all for the rest of the central nervous system, if the term is bold, then you want to know the name, you want to be able to identify the structure, you also want to know the function. Okay. If it's not bold, then it's just knowing the name, but you don't have to worry about function. So the homework I gave you guys, anyone start?
Yeah.
Oh, you did. Nice, impressive, very cool. So that is actually, basically, that's the stuff that you need to know the function for. So that's going to help you out, kind of like what I did with the muscles. You know, the ones that are bolded, those are what you had for homework. It's the same thing with the nervous system. So actually going through the structures on page 27, for part A, we'll start with cerebral cortex. Maybe I should ask you guys and see what you guys remember. Should I ask you guys? No.
Just tell us.
Too much, right? Cerebral cortex is going to be the outer portion. So cerebrum, we'll start with cerebrum. Cerebrum is going to be the larger part of the brain. So all of this is the cerebrum, and then this little part down here is the cerebellum. So cerebellum is a small portion that's like posterior and inferior to the occipital lobe, and then cerebrum is going to be this whole main portion of the brain. That's all of that. And then we have the cerebral cortex. So cerebral cortex is going to be this outer portion, because basically when you open this up, you'll actually see areas inside the brain that are going to be a little bit different in color. And so that's going to be like the medulla, or like the inner portion of the brain. So the cortex is going to be the outer layering, the outer portion. You also want to know longitudinal fissure. So what's a fissure?
Line.
A line, which is basically going to be like a true separation. So think of a fissure as a true separation. So we have the separation right here, and the way that it's traveling, it's basically going to be considered longitudinal. So this longitudinal fissure is going to separate the left and right hemispheres of the brain. We also have the transverse fissure. And so transverse fissure, if we take this towards the back, transverse, which direction is that going to go? Side to side, going across. Basically, this over here is like another true separation. So that's going to be between the cerebrum and the cerebellum. That's going to be the transverse fissure. So transverse fissure is going to be the separation, or the line between the cerebrum and the cerebellum. We also have sulcus. So what would a sulcus be? A groove. Yeah, so fissure is like a true separation. Sulcus is going to be just like a shallow groove. So we have the lateral sulcus and central sulcus. So lateral sulcus, we'll start with that one first. On the side over here, you guys see there's a groove right there. That's going to be the lateral sulcus. So on the sides where like the temporal lobe is, right above it, that's going to be your lateral sulcus. And then you also have a central sulcus, which is going to separate like the frontal and parietal lobes. And that's going to be going across this way. There's a bunch of little grooves, a bunch of sulci in here, but there's usually going to be one that goes more all the way across. Does this have it? But basically when you look at this, for instance, there's one that goes all the way through. So that's going to be your central sulcus.
That's the frontal and parietal separation.
And that's going to separate the frontal and parietal lobes. Does somebody want to turn that off? Nobody wants to.
It's not mine.
It's theirs. Okay. All right. So pre-central gyrus and post-central gyrus. Let me actually grab one that's color coordinated, just so you guys see it first and then memorize it.
Wait, so the central sulcus, it's not like the front? It's not this?
No.
It's this one?
That's just there so you can take it off.
We'll use this one for now. Since it's your first time seeing it, we'll use color, and then afterwards you want to learn without the color. So looking at this, we have like red and pink on one side, we have more of the blue shades on the other side. So this line right in between the red and blue, that's going to be your central sulcus. So that's separating the parietal and frontal lobes here. And then you have the red portion, which is more towards the front. So if this is in front of the central sulcus, then what is that called? Pre-central gyrus. So sulcus is going to be a groove. Gyrus is going to be the parts of the brain that stick out. So you can think of like mountains and valleys. So valleys are going to be the sulcus. mountains are going to be the gyrus, where it bulges out. So this is going to be the pre-central gyrus in red. And then the blue one back here, that's behind the central sulcus, so what's that going to be?
Post-central.
So post-central gyrus. So post versus pre, before and after. And then with function, you guys will go over that in your homework.
So, sorry, you were saying that the gyrus are all the little groups?
Yeah. So the grooves are going to be the sulcus, or sulci. Gyrus is going to be all the bumpy parts that stick out.
Yeah.
Frontal lobe, where's the frontal lobe?
Front.
Right here. So frontal lobe over here. What's on the side?
Correct.
Temporal.
Temporal.
Temporal over here. Parietal lobe is at the front over here. We have occipital lobe towards the back. And then there's one more lobe. Insula. And then do you guys remember how to find insula? On the inside. So what do you have to open up?
The temporal.
There you go. So the lateral sulcus over here, if we were to open that up, basically we have to separate the temporal lobe from the parietal lobe, and we can actually take out some of the frontal. Move that. And then we can start to see this little area right there that looks just like the cerebrum. So it's the same color, it still has those grooves and mountains, so the sulcus and the sulci and gyri. That's going to be the insula, right in there.
So that'd be deep to the frontal.
It's deep to, so it's more lateral, so it's going to be deep to the temporal and parietal lobes. So you basically separate those two, or open up at the lateral sulcus, right in there. That's going to be the insula. Also, you have basal nuclei. So when you take these apart, you will have to put them back together, so it is kind of like a puzzle. And sometimes people have a hard time with it, they struggle with it. One of the things I want to point out is, yeah, you have to like take little pieces apart in order to really get the core out. So what I'll do is I'll move things around a little bit to open this up. And then we get to this over here. You guys want to be really careful with this light blue structure, that is representing the ventricles, which are going to be the fluid-filled cavities inside of the brain. This is plastic and it breaks easily. So all of the models that we have, they are actually broken except for one that's still intact. And I think this is actually the one that is intact. So when we look at this, you can see it from the outside, but then the other part of it is actually inside of the other plastic part. So you'd want to really be careful when you are removing this. So like this portion you want to remove out, and then we can slowly start to open things up without breaking anything. And you guys are recording me, so I'm like, don't break it. So I got it up. Ventricles, right? So again, this is the only one that is still intact, so if you're having a hard time taking it out or putting it back in, let me know so that we don't break the one that's actually good. And the ones that are broken, you still want to be careful with them. We don't want to break them any further.
Are you saying the whole model is broken or just the ventricles?
What was that?
You were saying the whole models are broken or just
The ventricles.
Oh, okay.
These, yeah, because it's like embedded in certain regions, so people just kind of pull them apart and then it snaps and it breaks.
Okay.
So the reason why I took it apart right now is because I wanted to show you guys these red areas inside the brain. These are representing the basal nuclei. So we talked about these in lecture, and they have different names. You don't have to memorize the different names, but you just want to know in general that these are basal nuclei. So we see them in here, so it's like we have the insula on this side, and then deep to the insula we have some basal nuclei over here. Another example, for instance, with this portion, if we open this up, you see another example of a basal nuclei in here. So in the models, they're going to be like just a bright red color. So those are going to be basal nuclei. Okay. And again, for that, you just want to know general function for all of them together. And I have to put all this back. So we also have olfactory bulb and olfactory tract. So I'm going to use this one without taking, so this is going to take a while to put it back together. But say you have the whole brain, which is nice and put together, nothing has been moved. If we look at it from underneath, we have these white lines that are traveling out. Basically, that's going to sit kind of like where the ethmoid bone is from the top of the skull, right? So that's going to be your olfactory bulb and olfactory tract. So the tract is going to be the long portion, olfactory tract. And then the olfactory bulb is going to be the end, which is going to be more round or more oval-shaped. And then coming off of the olfactory bulb, there's going to be olfactory nerves. Tiny little nerves are going to come out of that. And those nerves, again, if it's sitting on the ethmoid bone, what is the name of that plate? Cribriform plate. And then what do you notice with that cribriform plate? It has a whole bunch of holes. So those holes are there so that the olfactory nerves travel through them. And that's going to be basically like the roof of the nasal cavity. Pretty cool. Next thing you have corpus callosum. Let's see, I'm actually going to use this model. So this one is nice and easy where you don't have to remove anything. It's just a sagittal view. Yeah. So we're looking at it from the side, and we see this white structure right there. That's going to be the corpus callosum or corpus callosum. Basically, you're going to have a collection of fibers or axons that are traveling across from one hemisphere to another hemisphere. So it's like communication between the left and right sides of the brain. So that's going to be the corpus callosum. Lateral ventricles, I can show you on, for instance, this blue structure right here. Where are the lateral ventricles? These two right here. So it's 1 and 2, right? Lateral ventricles are like 1 and 2. These over here. And then we have another model that's just like that. So notice there's like a lot of models to look at, and they all show kind of like the same stuff, but it's just like a different view of it. So you want to make sure that you go through all of these. Another model over here. So this actually looks like a spaceship. So with the spaceship, it's looking at the ventricles. So this is actually cavities. It's not a solid mass in the brain. This is just representing the actual space or the ventricles inside of the brain. And so here we have the lateral ventricles. And then we'll talk about the other ones when we get to that later on. You also want to know septum pellucidum. So which should I use? I'll use this one again. So going back to the half-head model, this white one I mentioned as the corpus callosum, and then this portion, which is number 5, right below it, that's the septum pellucidum. So that septum pellucidum is going to be, think of septum, a wall. That's going to be the wall that's separating the left and right ventricles. So it's like separating the 2 lateral ventricles. Okay, and that's going to be septum pellucidum. So for instance, if I were to ask you, on here, where would you find the septum pellucidum? Where would it be?
In between?
Right in between, right? So it's just basically like a sheet, like right over here, just a wall between the 2 ventricles. So what that means is, if we're looking at this model over here, in order to get to the lateral ventricle, we'll actually have to break through that septum pellucidum in order to get into that cavity. So there's a cavity on the other side of the septum pellucidum, and if we add the other half of the head model, then that's also going to have a cavity on the opposite side. Does that make sense? When you guys do the brain dissection, we'll actually be able to look at that. So you can actually kind of break through the wall and then see a little cavity.
So does each side have its own wall?
No, so it's just one wall.
combined?
It's one wall separating the 2. Yeah. So this is like off to the side a little bit.
Yeah.
So you're not directly cutting right into it. Yeah. Same thing with the nasal septum.
Mm-hmm.
Yeah. Next thing on here is choroid plexus. So on this model, the choroid plexus is this blue and red structure. So blue and red structure on here. Choroid plexus is a cluster of blood vessels and specialized cells that are going to work together to create cerebrospinal fluid, or CSF. So the blue and red right there, that's going to be the choroid plexus. So on this model, it's blue and red, which is like arteries and veins. But for instance, on the spaceship model for the ventricles, it's actually going to be pink in color. So the pink structures right in here, that's also representing the choroid plexus. So there's some between the ventricles, there's some in the 4th ventricle down here. Those are all going to be choroid plexus, which is going to produce cerebrospinal fluid. So it makes sense for it to be close to the ventricles because that's where the fluid's going to travel. Right. Let's see, fornix is the last one for that first portion. Go back to this one. And for this model, it's number 6. It's this other portion that's right below the septum pellucidum. So it's the white portion here, inferior to the septum pellucidum. And then again, in your homework, you'll go over functions. Are we okay so far? Yeah, you guys got this down, right? For part B, thalamus. There's 2 numbers on here. So basically this whole pink area is the thalamus. So all of this right here is the thalamus. It's basically like the center of the diencephalon. Do you guys remember I mentioned diencephalon in lecture? Diencephalon includes 3 major parts. There's the thalamus, hypothalamus, and hypothalamus. So all of that's going to be the thalamus. And then part of the thalamus, there's the interventricular foramen. So the interventricular foramen on here, let me find a better one. All right, this one doesn't show it very well. So I'll skip the number. So do you see number 11 with an arrow? So where the arrow is pointing to, that's going to be the interventricular foramen.
So it's not like the tip.
It's right at the end of the arrow. So it's like at the end of the arrow, right next to the choroid plexus, interventricular foramen. So it's like a hole between the 2 ventricles so that there's an even flow of cerebrospinal fluid going between the left and right sides. You guys see that? So number 11, and then above number 11 there's a little arrow.
That's where the
Interventricular
choroid
foramen.
plexus comes out of?
So you have the choroid plexus, so say we were looking at this over here with this model. You have the choroid plexus that we're looking at on the other model, and then you have the lateral ventricles. Basically, that right in the middle is going to be the interventricular foramen. So all of this is space, and the interventricular foramen is also going to be space. So that's going to be a space or like a hole or opening that's connecting these 2 so that the fluid is going to travel evenly between the 2. What was that?
Is it this hole you can see from the side?
No, that's going to be the intermediate mass, which is another structure we'll look at.
Oh yeah, I think I remember this.
So again, this is not a solid structure. This is all fluid filled. It's a cavity. It's a space. So the foramen is also going to be an empty space, which has fluid. So let me make sure you guys saw that too. So number 11 with an arrow, and then where the arrow is pointing, that's the interventricular foramen. So it's going to be a hole between the left and right lateral ventricles. And it's right by the choroid plexus. Other thing on here, third ventricle. So basically, like the space out here is going to be the third ventricle. So we talked about the thalamus is going to be like the lateral walls of the third ventricle. So the space or cavity out here is a third. And then if I were to use the spaceship model for that, that's going to be all of this over here. So that's going to be the third ventricle. So it's just 2 different views. Okay. So what would you find over on this side? What structure?
Thalamus.
Thalamus on this side, and then thalamus over on this side, and between the 2, you have that third ventricle. Okay, is that making sense?
So it's actually between 2.
The third ventricle is going to be between the left and right thalamuses. Thalami? Yeah. Next thing you have hypothalamus. Where would you find hypothalamus? Below the thalamus, so that's going to be like this area right there. So hypothalamus is going to be more inferior and anterior to the thalamus. So again, hypothalamus is inferior and anterior to the thalamus. Next thing you have is the pituitary gland, which is number 16 on this model. And so pituitary gland is going to produce and release a whole bunch of hormones that you guys will learn about later on this semester. So it's the pink structure.
That's the one that sits in the cellular.
Yes, 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 yes. Yay.
I was hoping that was going to be a question on the exam.
So number 16, again, that's the pituitary gland, which sits in the cell turcica. Of what bone?
Sphenoid.
Sphenoid, right? Infundibulum is basically the white connection between the hypothalamus and pituitary gland. That's going to be the infundibulum. So I'll show you on this right here, infundibulum, the connection between hypothalamus and pituitary gland.
The white part?
The white part right in there. And then there's also another model that I can show you guys on that. Do you guys want to see the other model, or are you guys okay with this?
Sure. Why
Should we
not?
grab it?
They're all gonna
Is
be
that
on the
supposed to
test.
be funny?
No, it's all gonna end up on the test. Everyone's gonna.
So again, so many different models, right?
Get it?
Let me take this one out. I'm making such a mess. I won't use this one. So we'll go back to this half over here. So with this model, for instance, it looks different. It's not as colored as the other one that we saw earlier. So like this is representing the thalamus. You have the hypothalamus, pituitary gland, and then that line right there is the infundibula. Okay. So thalamus, hypothalamus, infundibulum, pituitary gland. Do you see how it's a different view?
Can you do that one more time?
Thalamus, hypothalamus, infundibulum, pituitary gland. Does it make more sense to look at this model?
Yeah.
Yeah. Okay.
Is that like a vessel? That goes to your-
Which one? This right here?
Yeah.
It's connective tissue.
Oh,
Yeah. So you guys will learn more about that later on. You'll learn more about that later on when we get into the endocrine system. Next on your list, what do we have? Mammillary bodies. Go back to this one over here. So back to this brain. There's 2 green dots on here. Those are the mammillary bodies. So 2 green dots. Which are mammillary bodies.
Do they have numbers?
Not this one. So don't memorize color, because here it's green, but I think another model is like yellow in color. So those 2, it's going to be a pair. So those are mammillary bodies. And then you have the pineal gland. And so pineal gland, I'll use this one. On the half head model, it's number 14. So we have that right there. That's going to be part of the epithalamus. So it's like posterior to the thalamus. And that's going to be pineal gland. Pineal gland is going to play a role in your sleep, wake cycles, circadian rhythm, day and night cycles. Produces melatonin. Give me guys some answers. So it's going to be pineal gland. And then you have the intermediate mass. So that's number 8 on the thalamus for the half head model. It's brown in color. So that's an actual structure.
So it's in the middle.
Right in the middle, yeah. So intermediate mass. Intermediate, it's basically between the left and right parts of the thalamus. So that's going to be like the connection point between the 2 halves of the thalamus. And that's going to be the intermediate mass. Does that make sense? So for instance, if we go to the spaceship again, and we see a little hole right in there, that hole is there so that the intermediate mass can travel through it to connect the left and right halves of the thalamus. Does that make sense?
Basically surrounded.
Which is, what was that?
I said it's basically surrounded.
And it's going to be surrounded by fluid, yeah. So it's a connection between the 2 halves of the thalamus. Okay. So many things going on. Let's see. Okay. Which one should I use? Trying to avoid taking this one apart. Alright, I'll do this. I'm going to cheat a little bit. So going back to this little area over here is part of the brainstem. So this is actually the same as this over here. So you have like a big round portion, which is like a big bump in the front. Then we have the same thing going on this side. This is just a little bit bigger, but I will stick with this first and then use this one to show you what the last few things are for this, for part B. So for part B, in bold, there's optic nerve, optic chiasm, and then optic tract, which is all dealing with vision. So if I hold this up this way so you can see it like underneath, kind of like below the brainstem area, there's these ridges that are traveling up right here and right here. Those are going to be the first thing on there, which is the optic tract, the third one. So optic tract are the ones that are traveling over here. They're kind of like inferior to the thalamus, and they're going all the way across. So they're like extending out anteriorly. So those are going to be the optic tracts. Okay. If we use this model, for instance, Same thing if we have it like this and we hold it this way so we can look underneath. There's these that are in orange. Those are going to be optic tracts as well. Okay with that? So both sides, left and right, optic tracts. When the optic tracts come together, they basically cross over, and the area where they cross over is optic chiasm. So that's going to be like the X portion, where they meet and cross.
Is that like the same thing that you were talking about last week about left brain doing the right side and the right brain?
Yes.
Okay, so that's what it is.
So this is with vision, yeah.
Same,
So
same concept.
exact same thing, yeah. So like if you're looking through one eye, you cover the other one, both hemispheres are both going to receive the same information from one eye because of the optic chiasm. So you have axons that are traveling and staying in the same hemisphere, but you also have axons that are traveling over to the other side for that bilateralization, basically. Yeah. And then the last part on here, where it extends anteriorly, that's going to be then the optic nerve. So optic nerve is where it extends out anteriorly after the crossover. And so this is the part where it's actually going to extend out to the eyeballs to receive the information. Okay? Okay, so now we can move on to the midbrain. So midbrain, looking at this model for instance, we can see a couple of things. So this whole area right here is going to be the midbrain. The first thing on your list is the cerebral aqueduct. So cerebral aqueduct is this little tunnel right in there, which is basically going to be the connection between the third and fourth ventricles. So cerebral aqueduct is a little tube-like structure inside of the midbrain, which is going to be the connection between the third and fourth ventricles. So it's like a continuation of where the cerebrospinal fluid is going to travel to get between third and fourth. Do you see the little opening right there? You can also see the cerebral aqueduct on the spaceship. So with this over here, if we said this is the third ventricle and if this is the fourth ventricle, then where's the cerebral aqueduct? There you go. So it's going to be that tube. So again, this is an empty space. So it's a tube that's connecting between third and fourth. Okay? So apparently we have spaceships and aliens in us.
In our brain.
And I always have this urge to like have like going out of my on my stomach or something, just like alien. So that was cerebral aqueduct, and then you have corpus quadrigemina. What is corpus quadrigemina? The four bumps. So the two pairs of twins, right? So on this one, it's kind of like the picture where you only see half of it. So we have a bump at the top and a bump at the bottom, but it's going to be paired. So you have like a left and right top bumps, left and right lower bumps down here. So corpus quadrigemina would be all four, but if we're just looking at the top pair of bumps, that's going to be the superior colliculi. The bottom pair of bumps, that's going to be the inferior colliculi. So these are going to be posterior to the, so it's part of the midbrain, yeah, but it's going to be on the posterior side. So the way that's positioned is like this is anterior and this is posterior.
Which one's the bottom side?
So top, superior colliculi, below that is going to be inferior colliculi.
Thank you.
And then cerebral peduncles, that's going to be anterior. So flip this around, so look at the anterior side of the midbrain. That's going to be these, where you can see like fibers going up and down, so there's lines going up and down. That's going to be the cerebral peduncle. So you're going to have tracts there, or axons, that's going to communicate between the higher brain centers and the lower centers. So I'll move this and this. So it's bilateral as well on both sides. Questions so far? No? So inferior to the midbrain, then this large bump right here is the pons. And so with the pons, that's going to be anterior to the cerebellum. So basically, if we look at this again over here, we see the big bump in the front. What's that called? Pons. And then posterior to the pons over here is then going to be the cerebellum. With the cerebellum, there's a couple of features. I'll use this one. This is basically going to sit like that. So with the cerebellum, you need to know the middle cerebellar peduncle, which is going to connect to the pons. So basically, if we're like matching these two up over here, that's going to sit like this and like this. And then the pons is going to be anterior to it. So for this over here, the areas where they have the little dots, that's kind of like where you're going to find the middle cerebellar peduncle. So it's going to be the connection point between the cerebellum and the pons. So it's just the areas over here and over here. It's not really well depicted on here, but it's basically going to be the connection point between that and the pons. Folia. So you have folia that you need to know. There's Arborvitae, which is tree of life, which I'll use this one for that. And then you also have Vermis. So what I will use for this, I'll go with the Vermis first on this one. So Vermis, basically when you look at this, you guys see there's like it kind of looks like an additional structure that's kind of like in between the two lobes over here. So that's going to be the Vermis. So think of like, Vermis is like a worm. Like appendix is the vermiform appendix, so vermiform or vermis is referring to a worm. So it's like a worm-like structure that's between the two lobes of the cerebellum. So that's going to be the vermis. And depending on which model you look at, some of these would be better than others. So if I use the half-head model, do you guys see the white lines inside the cerebellum? That's going to be the tree of life, but you want to remember the name Arborvitae. So Arborvitae is going to be the white lines inside of the cerebellum. So it's basically like the white matter inside of the cerebellum. All of the white branches looks like trees. That's going to be Arborvitae. So on the exam, actually write in and memorize Arborvitae, not just put tree of life. I've had students that just put tree of life, and that's, you want to go with the actual name for that. Fourth ventricle. So if you look at the fourth ventricle, if we go back to this over here, that's the last portion right there, which is also going to have the cerebrospinal fluid, which is going to be down here. And then, getting closer to the end, I want to make sure we have time for exam stuff. With the medulla oblongata.
Do we do the folia?
Folia are going to be like little ridges. So like all of the ridges on here, that's all going to be folia. So like with the cerebrum, you have the gyrus, the gyri, and the sulci. With the cerebellum, you have these other ridges, which is referred to as the folia.
Okay.
And then, where was I?
Medulla.
Medulla. I'll go with this one first. So medulla oblongata is this over here. And I think I mentioned that whenever I say that, I always think of Adam Sandler.
Yeah.
So with the medulla oblongata, off to the sides, like the lateral portions of the medulla oblongata, there's like oval-shaped structures. Those are going to be olives.
The little dots.
It's like an oval, this
Oh, the
right
bump.
here, the bump.
Big bump.
So an oval-shaped bump, yeah. That's going to be the olive. So more lateral, oval-shaped bump as the olive. It's not colored on this one, but if I grab this smaller model, the pink over here, that's going to be the olive. So pink, oval bump on the lateral sides of the medulla oblongata, those are going to be the olives. And so there's left and right. And then more anterior on the medulla oblongata, these two bumps over here towards the front, those are going to be the medullary pyramids. So two over here towards the front. Those are going to be the medullary pyramids. They're longer. You actually have axons that are crossing over in this area, so you have decussation of fibers, so crossing over of axons. So it's these two in the front. And then the last few things, meninges. The one that I haven't used yet. This is such a mess. We only have, I think we only have one of these. And so this is the only model that shows the meninges. All the other brain models don't have the meninges. So meninges are going to be layers of connective tissue that are going to surround the brain and the spinal cord. So it's like a form of protection. And there's actually going to be 3 layers. So first thing on here, let's see. I'll go over the layers first and then I'll talk about the detailed stuff in here. But the first layer over here, this thicker portion that's more superficial, that's the dura mater. So dura mater is the thickest layer of the meninges, and it's the most superficial. So it's these two over here. And it's going to go all the way across. It's going to cover the whole thing up. If we peel off the dura mater, then the next layer is going to be this, which is the arachnoid mater. Arachnoid mater is, it's also connective tissue. It's a little bit more loose than the dura mater. And so we'll look at this in lecture and I'll talk about it a little bit more. And if we were to peel off the dura mater, then we get to a third layer, which is going to be the pia mater. So pia mater is going to directly attach to the brain. And it's also going to like follow into those grooves, like all the sulcus or the sulci. It's going to go actually like lining up with all of that. And so that's going to be like the third layer that directly attaches to the brain. If you try to peel off the pia mater, you're actually going to start peeling off the neural tissue. So it directly adheres to it. Think of it as like a visceral layer. And then with the details under dura mater, there's falx cerebri and tentorum cerebella. So falx cerebri, I'll just read this up. If we remove this right here, you have all of those three layers that's forming this like wall, kind of like a septum between the left and right hemispheres. That's going to be the falx cerebri. So falx cerebri is going to be, again, a continuation of the meninges that's going to travel into the longitudinal fissure that separates the left and right hemispheres of the brain. So you want to make sure it's like you're really protecting all the parts of the brain. So it's also going to go into that little opening between the hemispheres. And then with the other fissure that we have, that's going to be where you have the tentorum cerebelli. This model doesn't really show it. But basically, here you have the cerebrum and the cerebellum. So like right in between the two is where you'd find the tentorium cerebelli. So it's going to be more of that, the meninges, that's going to travel into that transverse fissure to basically line up the separation between the cerebrum and the cerebellum. So it'd be right in between the two. Questions so far? What's next on your list?
Subarachnoid space.
So with the dura venous sinuses, it says these will be reviewed in the vascular system. So we'll look at this in more detail, but with this model we do see one of them. So this top one right here is the superior sagittal sinus. So superior sagittal sinus is going to be formed by the separation of the dura mater. So dura mater is actually two meningeal layers that are fused together. And then there's a certain region where they separate to create this dural sinus. So as a dural sinus, this is where you're going to have blood that's going to leave the brain to go into the dural sinus. Eventually it's going to go through other parts within the brain region, over to the jugular vein, and back towards the heart. So that's the superior sagittal sinus.
So leaves brain.
And then inferior sagittal sinus, that's the one that's below it. So superior sagittal sinus at the top, and then inferior sagittal sinus is right below it. So we have these two. And then there's plenty more that you guys will see later on when we get into the cardiovascular system.
Subarachnoid space.
Okay. With the subarachnoid space, doesn't really show up very well on here. Basically, subarachnoid space, if we were to remove the arachnoid mater, there's going to be like a little space right below it. That's where you're also going to have cerebrospinal fluid. So you have cerebrospinal fluid in the ventricles inside the brain. You're also going to have cerebrospinal fluid that's surrounding the entire brain on the outside, but it's going to be deep to the arachnoid mater. But we don't really see it here, so it's actually going to be deep to this layer. And the last thing here is arachnoid granulations or arachnoid villi. That's going to be all of these little white dots. So all little white dots in here and in here. And then we'll talk about the arachnoid villi in lecture. It'll make more sense when I actually see it as a picture. So all the little white dots, right there. Basically, it's going to be like kind of like a pathway for the cerebrospinal fluid to leave the brain area and then go into the jugular sinuses to basically go back into the bloodstream. So white little dots. Questions?